The decay required practical is really another version of the enzymes required practical that came about in Unit B2. In the enzymes required practical, you had to investigate the effect of pH. For this required practical, you're really just investigating the effect of temperature on enzyme activity. What do we mean by that? This is about decay, and we need to remember that decay happens by microorganisms. And what the microorganisms do, they don't have a mouth to get the food into their body. They actually release enzymes out of their cells and onto the organic matter that's going to decay dead organisms' waste. So they release enzymes. And it's those enzymes that are going to be doing the work. So a higher rate of decay just means it's a higher rate of enzyme activity. Slower decay means that the enzyme activity is lower. So when you are thinking about the decay required practical, really what you should be thinking about is enzymes. Let's have a look at some of the key words from enzymes. Enzymes are proteins. They are biological catalysts. They have an active site that the substrate fits into. So the sort of go-to for everybody is the idea of the Pac-Man as the enzyme. The active site is the mouth. And the substrate for that enzyme has to be the correct shape to fit into the mouth. So you've got the active site, we've got the substrate. When they come together, you get what's known as the enzyme substrate complex. We need to know that the active site and the substrate fit together and that they have a specific or complementary shape. So again, the idea that unless the substrate fits the active site perfectly or close to perfectly, the reaction is not going to occur. In terms of enzymes, we also need to think about what happens to the enzymes when the pH or the temperature changes. And this is going to be a really important word that we're going to come back to when we discuss the practical itself, which is that the enzymes can denature. Denature means that the active site changes shape. Why does it change shape? Because the bonds holding the protein together start to break. So instead of our nice Pac-Man, we're going to end up with a misshapen protein, and now the substrate doesn't fit, so there's no reaction that can happen. And when we talk about the decay required practical, an easy way to think about it is it's just an enzyme required practical. Last thing before we move on to the practical itself is that there are two things that can cause an enzyme to denature. There is pH. If the pH is too low, if it's too acidic, or if it's too high, too alkaline, the enzyme will denature. And then, of course, you have temperature. If the temperature is too high, the enzyme will denature. The basic equipment list for the decay required practical is the following. You've got full fat milk, you've got lipase, water bath, sodium carbonate solution, phenolphthalein, glass rod, boiling tubes, pipette syringe, stopwatch, and thermometer. So let's talk through what each of these things are doing. The milk, the full fat milk, is going to be a source of the lipids for this experiment, and lipase is an enzyme that will digest the lipids. And the lipids, as you know from Unit B2, are going to break down into two things. They're going to break down into glycerol and into fatty acids. If we remember, the glycerol is the rectangle, the fatty acids are the hairs coming off, so we like to remember of lipids as a hairy rectangle. The key thing here is going to be the fatty acids, with the key bit here being it is an acid. So this is going to lower the pH of the solution. You're going to start with a lipid. As the lipid gets broken down by the enzyme lipase, it's going to produce fatty acids, and that's going to lower the pH. Why do we have sodium carbonate? We're using the sodium carbonate as a source of raising the pH. It's alkaline. So this is going to initially raise the pH to, around 10, 11, something like that. And then as the lipase digests the fatty acids, we're going to lower that pH back down. Why do we want to do that? Because we're using phenolphthalein as an indicator. Phenolphthalein is pink above around pH 8. So we're going to add the phenolphthalein and sodium carbonate and the milk and the lipase all together. The phenolphthalein, due to the sodium carbonate, is going to make the solution turn pink. And then as it gets down to around pH 8, it's then going to turn colorless. So that's how we know that the enzyme is fully broken down. Our lipids is we're using the phenolphthalein as a qualitative measurement there, as a way of telling when the pH is being lowered. The faster that pink color disappears, the higher the enzyme activity, the faster the lipase is breaking down the lipids, and therefore the higher the rate of decay. The rest of the equipment there is pretty straightforward. Really, really important when we think about a water bath that we say that it controls the temperature of the water. A lot of people, when they look at these questions, they say it's the thermometer that controls the temperature. Of course, it's not. It's the water bath that we're using to control the temperature. So if we're looking at the effective temperature on the rate of decay, temperatures are independent variable, so we need to be able to control that so we can try out a bunch of different temperatures. Sometimes you'll see people doing it in a beaker with different temperatures of water. That's fine. It's not going to give you as accurate measurements. You're going to want to do it with a water bath to get the most accurate measurements. Glass rod is just to stir. The boiling tubes is to hold your sodium carbonate, phenolphthalein, and milk in one, and then your lipase in another. The pipette or syringe is to add the sodium carbonate, add the milk, and add the lipase so that you can get accurate measurements. It's possible that they say the student used a pipette. What's a more accurate way of measuring it out? A syringe is going to give you a more accurate measurement than a pipette. Stopwatch is to time how long it takes to go colorless. And the thermometer is just to check or verify the temperature. So possible questions that they can ask here is, why did they use sodium carbonate? And the reason that you're using the sodium carbonate is to initially raise the pH to make your phenolphthalein turn pink. If you didn't have the sodium carbonate or another alkaline to add in there, you're not going to get that pink color to start with. And what you're looking for is when that pink color disappears. Why do we want full fat milk? That might be a question. Well, the lipids are the fats. So the higher the fat content of the milk, the more lipids there are going to be for the lipase, for the enzyme to work on. If you were using, the red milk from Sainsbury's or something that has a really low fat content, your reaction is going to happen a lot slower because there's not going to be enough lipids for the lipase active site to collide with. For the actual experiment, what you're going to be doing is testing different temperatures. And ideally you would do each one 3 times. That might be a question. A student does this experiment, but they don't do it 3 times. They only do it once each. How could they get more accurate or reliable results? You would say they should do the experiment 3 times, check for outliers, calculate a mean to get more accurate results. When you're testing for the different temperatures, a really important step here, just like for the paper 1B2 effect of pH on enzyme activity, is you're going to want to set up 2 different boiling tubes. In the one boiling tube, you're going to have the lipase. In the other boiling tube, you're going to have your milk, your sodium carbonate, and your phenolphthalein. It's possible that they ask you about a different indicator. But almost definitely they'll ask you about phenolphthalein. You're then going to leave those in the water bath for 5 minutes, or at least until with your thermometer you check that they're both at the same temperature. They really, really like to ask the question, why did you leave the 2 boiling tubes in the water bath before mixing them together? There won't be any reaction here because the lipase is in a separate thing from the milk, so the lipids can't be broken down. And the reason is, the only answer that you need to give if they ask you why do you keep them for 5 minutes is so that they can get to the same temperature. It's that easy. Those 2 words will get you full marks. It's just 1 mark, but it'll get you the full mark on that question. So watch out for that question there. What you're going to do after you add it in is you're literally just waiting for the color to change and writing down the time that takes on your stopwatch. A trick here that they might get you at, it's unlikely, but they might get you at here, is we say that the phenolphthalein goes from pink to colorless. But it's the phenolphthalein that goes colorless. You still have the milk. So if they ask you what the appearance is, it would be white or cloudy. Don't say that the solution itself goes colorless, because it's just the phenolphthalein does. So one way to think about this is it goes from pink milk to normal milk. Normal milk is not colorless. It's not see-through, at least not any milk that I've had recently. So watch out for that as well. Other than that, it's a pretty straightforward thing here. We'll get to the interpretation of the data in a minute, but first let's talk about how, if they ask you to increase the accuracy, it's a nice question for any of the required practicals. How could you increase the accuracy? They'll give you a situation with some students. What could you do to increase the accuracy? The first thing you should always look for is, can they do more repeats? So did they only do 1 repeat? And then also add that you're going to calculate the mean. If they use the word apparatus, there's 2 different things that you can say here. The first is use a syringe instead of a pipette, because you'll get more accurate volumes for what you're adding in. The second is, you don't need to do color change. You're really just looking for a change in pH at an arbitrary level here. So what you could use is a pH probe instead of the indicator. So instead of looking for a color change, the pH probe just reads out a number. So it displays a digital number, and maybe you're looking for it to go to pH 8. As soon as it gets to pH 8, you stop the stopwatch. The reason that that's more accurate is it is a quantitative, not qualitative judgment. If you're looking for a color change, you and the person you're doing the experiment with might argue about, oh, is it still pink? It's a little bit pink. I think that's clear. No, I don't. So different people can have different results. If you're looking at a number, when it turns to 8, nobody's going to argue that, no, I think that that's actually 7.9 or something like that. If it says it's 8, it's 8. Last but not least here, we need to consider how you're going to interpret the data. And because it's an enzyme practical, there's 2 different ways that they can display the data that will be exactly the same. Okay? If we have our data displayed as graphs, on both the x-axes, you're going to have temperature. If the y-axis is time to go colorless, your graph is going to look like this. If, on the other hand, it is rate or enzyme activity, so rate of decay, the graph's going to look like this. And the reason for that is that rate is literally just 1 over time. So the graphs are going to be inverse. Okay? So if you see time on the y-axis, what you need to be thinking is the shortest amount of time, that's where the decay is happening best. That's where the enzyme activity is happening best, because you're getting that color change in the shortest period of time. If, however, it says rate of decay or enzyme activity, then the highest rate is the best one. That's the optimum, to put it in enzyme terms. Okay? Same thing with the data table. Whatever is in the first axis will be the temperature, and whatever's in the second axis, or the second column, I should say, or third or fourth, if you're doing repeats, will be either time or rate of decay or enzyme activity. So have a look out for that. If it's time, shortest time is the best. If it is rate of decay or enzyme activity, then the highest number is the best. The last thing to say here then is that you always need to have in the back of your mind when you're doing these questions that it is an enzyme question. So if we have rate of decay on the y-axis and temperature on the x-axis, and we have our nice graph that goes like that, the best one, the optimal point, the highest rate of decay, is going to be up here. That's our optimum. On the right-hand side of the graph, the reason that it goes down so sharply and ends up being going down to nothing is that over here, the enzymes, the lipase, denature. Okay? And going back to the very beginning, if the enzyme denatures, then that means that the substrate, which in this case is the lipids, can't fit the active site anymore, which means that you're no longer going to have any successful collisions. You're not going to have the enzyme breaking down the lipase anymore. So on the right-hand side, if you had numbers that were going up, higher and higher rate of decay, but then say the temperature went up to 50 degrees or 60 degrees or something like that, and suddenly the numbers dropped off, those aren't outliers. They're trying to catch you out there. That's where the enzyme has denatured. This would be a lovely place for them to bring in paper 1 stuff, which they're allowed to do on paper 2, is to increase the rate as the temperature goes up and then have it suddenly drop off. That will be what separates the really smart students from the superstar students, so you need to watch out for that. On the left-hand side of the graph, if they want to be really, really mean, it doesn't denature. Denaturing on the left-hand side happens on the pH graph. So here, it's not denaturing. What's happening here is as you're increasing the temperature, so as the temperature increases, you're actually giving all the particles, you're giving the lipids and you're giving the enzymes more kinetic energy, which means that there's going to be more collisions just because they're moving around fast. There's going to be more collisions between the lipids and lipase. So you're going to have a higher rate of decay. And that goes all the way up to where that optimum point is. And then past that, the enzymes start to denature. And remember, the denaturing means that the bonds that are holding the protein together, the enzyme as a protein, start to break, which means that the active site changes shape, which means that the substrate no longer fits. Okay, so if they want to bring in some of the paper 1 stuff, and they're entirely allowed to do that, if they're looking to be mean on this question, then that's where you're going to go. Good luck if you get one of these questions. If you remember that it's an enzyme question and you use the enzyme keywords, I think that you're going to do great.